Hello everyone. Myself Sapna Chaudhar, working as an assistant professor at IIMT College of Pharmacy, Greater Noida. So, firstly, we are going to discuss about the carbohydrate metabolism in today's lecture. So, what are the carbohydrates? Firstly, we are discuss about the carbohydrates. Carbohydrates are simply called as sugar molecules like glucose, fructose. These are sugar molecules are included in carbohydrates. There are types of sugars like reducing sugar, reducing sugar are also included in this category. So today we are going to discuss about the carbohydrate metabolism. If we are dealing with the carbohydrate, lots of types of carbohydrates are there, like monosaccharides, disaccharides, trisaccharide, and oligosaccharide. If more than 5 sugar molecules are in carbohydrates, are simply called as polysaccharide. So, in case of examples of monosaccharide, there are lots of examples of monosaccharides, but like the very typical example like aldolase and glucose are the monosaccharides molecule. Then ketose and fructose are simply monosaccharide molecule. In case of disaccharide, there are reducing sugar or non-reducing sugar. Reducing sugar like maltose and lactose, and non-reducing is like sucrose. In case of polysaccharide, it is again sub-classified into homopolysaccharide and heteropolysaccharide. In case of homopolysaccharide, starch and dextrin are the some common examples. And in case of heteropolysaccharide, hyaluronic acid is a common example of heteropolysaccharides. So the what is the carbohydrate metabolism? Simply carbohydrate metabolism, it is fundamental biochemical procedure that ensures the constant supply of energy to the living cell. If we see the energy provided during carbohydrate metabolism, it is simply in the form of ATP or in the form of NADH or in the form of FADH. So the main purpose of carbohydrate metabolism is to yield energy. It is lots of pathways where takes place during carbohydrate metabolism, like your glycolysis cycle and Krebs cycle are also included in carbohydrate metabolism. So the main purpose of glycolysis cycle is glucose is breakdown into the pyruvate or lactate. It is or after glycolysis cycle, that carbohydrate molecule is that pyruvate molecule is goes into the Krebs cycle, and through the oxidative phosphorylation it generates the ATP, which is required for day-to-day work life. So what is glycolysis? Glycolysis is the process in which glucose is broken down to produce energy. When we need the energy, carbohydrate metabolism is takes place. It produces two molecules. Like in case of firstly, we are dealing with the glycolysis cycle. In case of glycolysis cycle, simply a glucose molecule is metabolized up to the pyruvate. Lots of steps are takes place in glycolysis cycle, like glucose is converted into glucose 6 phosphate, and up to the glucose is converted into pyruvate. Lots of enzymes are also catalyze the reactions, and lots of intermediates were also formed in the glycolysis cycle. So in case of intermediates like glucose 6 phosphate and fructose 6 phosphate are also intermediates of glycolysis cycle. These are the main basic examples which are also help in HMP shunt or in Krebs cycle also. Pyruvate is going into the Krebs cycle also. So the basic significance of glycolysis pathway are in case of intermediates, that intermediates are also help in another cycles also. So in stage 1, like this diagram explain about the glucose when converted into pyruvate. That procedure is simply called as glycolysis, which is shown by the blue color. And in case of pyruvate is converted into again glucose. It is exactly reversible procedure of glycolysis, are simply called as gluconeogenesis. And in case of non-carbohydrate molecule is converted into the carbohydrate, like sugar molecule, glycogen is converted into glucose. That procedure is simply called as glycogenolysis. And in case of glucose 6 phosphate is converted into glycogen, that procedure is simply called as glycogenesis. So lots of cycles are there for the carbohydrate metabolism. So this is the cycle of carbohydrate metabolism, which is simply called as glycolysis cycle. There are total 10 steps where takes place in glycolysis cycle. First is glucose is converted into the glucose 6 phosphate. A single phosphate group is added to glucose. This cycle takes place in the cell cytoplasm. So one phosphate group is added to glucose in the cell cytoplasm, and the enzyme which is catalyzed this reaction is hexokinase. In this stage, a phosphate group is transferred from ATP to glucose, forming a glucose 6-phosphate. Simply you can call it as ATP is converted into ADP. So this glycolysis cycle is divided into two parts. Like first part, in that part, energy consumption phase is there, and in case of second part, energy synthesis that takes place. In stage 2, that is glucose 6-phosphate is converted into fructose 6-phosphate. It is a the enzyme is catalyzed. This reaction is phosphoglucomutase. In case of stage 3, phosphofructokinase enzyme transfer the fructose 6-phosphate to fructose 1,6-bisphosphate, where another ATP molecule is converted into ADP molecule. So the two ATP were used during first stage of glycolysis cycle. In case of this stage, fructose 6-phosphate convert to fructose 1,6-bisphosphate, and the enzyme catalyzed this reaction is phosphofructokinase. Simply, in case of stage 4, aldolase enzyme converted fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate, that and dihydroxyacetone phosphate, which is isomers of each other. These glyceraldehyde 3-phosphate and dihydroxyacetone phosphate are isomers of each other, and single conversion of this takes place, reversible conversion of DHAP and GLA takes place with the help of enzyme phosphate isomerase. And in case of this, we can consider it as a stage 5. Triose phosphatase isomerase convert dihydroxyacetone phosphate to the glyceraldehyde 3-phosphate. In case of stage 6, this undergoes two reactions, like the enzyme glyceraldehyde 3-phosphate dehydrogenase transfer one hydrogen molecule, glyceraldehyde 3-phosphate dehydrogenase transfer one hydrogen molecule to form glyceraldehyde phosphate nicotinamide. Adenine like NADH were formed during this procedure, and glyceraldehyde 3-phosphate dehydrogenase added a phosphate to oxidize glyceraldehyde 3-phosphate to form 1,3-bisphosphoglycerate. So basically, simply we can call it as a formation of two NADH molecule that takes place during the formation of 1,3-bisphosphoglycerate. And in case of 7th stage, phosphate is transferred from 1,3-bisphosphoglycerate to ADP to form two molecule of ATPs, and it forms the 3-phosphoglycerate. In case of and the enzyme which catalyze this reaction is phosphoglycerate kinase. In case of stage 8, 3-phosphoglycerate is again converted into 2-phosphoglycerate with the help of enzyme phosphoglyceromutase. And in during the formation of phosphoenol pyruvate, two water molecules are form two water molecules, form conversion of 2-phosphoglycerate to phosphoenol pyruvate. And the last stage of glycolysis cycle where takes place during the formation of pyruvate from glucose molecule, it yields 2 ATP molecule with the help of enzyme pyruvate kinase. So there are total 10 steps of glycolysis cycle takes place, but we can consider it as a main important part of carbohydrate metabolism. So there are some key points of glycolysis cycle we can discuss about it. It is the process in which a glucose molecule is broken down into the molecule of pyruvate. This is the main purpose of carbohydrate metabolism. Simply glucose metabolized to pyruvate, and then pyruvate goes into the Krebs cycle and form the ATP molecule. Total 32 ATP molecules were formed during metabolism of one glucose molecule. So the process takes place into the cell cytoplasm of the, takes place in cell, plant cell and animal cell also. And total 10 enzymes were used. So basically 2 to 3 enzymes are repetitive. So the total 6 main enzymes were are important in this procedure. The end product yields what? ATP molecule, NADH molecule during the process of glycolysis cycle. So the significance of glycolysis cycle, In case of glucose 6-phosphate, which is intermediate form during glycolysis cycle, first intermediate form during glycolysis cycle. It helps in HMP pathway also and glycogen synthesis also. So this is the main intermediate which is formed during the glycolysis cycle. In case of fructose 6-phosphate, which is second intermediate form in glycolysis cycle, it is also required for synthesis of pentose or triose like glyceraldehyde 3-phosphate. It is used in synthesis of glycogen also and pentose also. So this is the main pathway of glucose metabolism, and the end product of glycolysis is pyruvate, which is act as an intermediate of various pathways like gluconeogenesis and fermentation procedure. It is also used in fermentation procedure also. So the like lactic acid formation and ethanol fermentation, this pyruvate which is also help in this procedure. And if we are going to discuss about the importance of glycolysis cycle, it is the when our body required the energy, most of energy provided by the glucose metabolism, because after metabolism of glucose molecule, lots of energy is released which we can use for our day-to-day life, and it is the pathway that is used to convert the glucose into the energy. So this is all about the glycolysis cycle. And in case of second cycle, it is the cycle simply called as TCA cycle. TCA cycle, another name for TCA cycle is Krebs cycle also, or citric acid cycle also. In this cycle, firstly, pyruvate is converted into with the help of coenzyme. Pyruvate is simply converted into the citrate, and with the help of enzyme citrate synthetase, it is one water molecule. When citrate is converted into isocitrate, it forms the one water molecule, and the enzyme catalyze this reaction is aconitase. Like after the conversion of citrate to isocitrate, these two are the isomers of each other. So, in some articles, total 8 steps are shown. And in case of isocitrate, it's converted into alpha ketoglutarate. The synthesis of energy where takes place after this step, like when isocitrate converted into alpha ketoglutarate, energy released in the form of NADH with the help of enzyme isocitrate dehydrogenase. After the formation of alpha ketoglutarate, with the help of another coenzyme, it converted into succinyl-CoA, and the enzyme which is catalyzed this reaction is alpha ketoglutarate dehydrogenase. It also releases one NADH molecule. Like we are discussed earlier, one NADH is in 3 ATP molecules. So basically 2 NADH molecules are formed during this procedure. So total 6 ATPs were formed in second or third stage of TCA cycle. After conversion of succinyl-CoA to succinate, one coenzyme is help, and also succinyl-CoA synthetase enzyme is also catalyze this reaction. One GTP molecule is formed during this procedure, and when succinate converted into fumarate, one FADH, two molecules of FADH are formed with the help of enzyme succinate dehydrogenase. Fumarate is converted into malate with the help of enzyme fumarase. One water molecule is also added during this reaction, and after conversion of malate to oxaloacetate, one NADH is also formed with the help of enzyme malate dehydrogenase. And the last stage of TCA cycle is oxaloacetate is converted into again citrate with the help of one water molecule, and enzyme catalyze this reaction is citrate synthetase. So basically lots of stage are also in the TCA cycle also. TCA cycle are simply called as Krebs cycle or citric acid cycle. So various forms of energies are produced during TCA cycle. Like in case of glycolysis cycle, energy is released only in the form of ATP. But in case of TCA cycle, NADH is also formed, FADH2 is also formed, and GTP is also formed. So total 24 molecules of ATPs are formed during the TCA cycle. So in case of a single turn of cycle, 2 carbon enter into the acetyl CoA and 2 molecule of carbon dioxide are released. 3 molecule of NADH2 and 1 molecule of FADH2 are generated, and 1 molecule of ATP and 1 molecule of GTP is also converted. Steps of TCA cycle. This figure, glucose is simply produces the pyruvate, but 2 pyruvate molecules are released during the glycolysis cycle. So 1 pyruvate molecule is 12 ATP. So 2 pyruvate molecule is 24 ATP. 24 ATPs are during the Krebs cycle, and total 8 ATPs were generated during the glycolysis cycle. So after metabolism of single glucose molecule, total 32 ATP molecules are generated. So this is the another cycle for glucose metabolism, like glyconeogenesis. Actually, this is not about the glucose metabolism, but when conversion of non-carbohydrate, non-glucose molecule is into the glucose are simply called as gluconeogenesis. The main precursor used for gluconeogenesis cycle are lactate, pyruvate, glucogenic amino acids, and glycerols are some common example of gluconeogenesis. During the starvation, our body required lots of energy. So to generate the energy in the form of ATP, glycogen is simply converted into glucose, and then that glucose is the energy which can use during starvation for our day-to-day work. So in case of glycolysis cycle, glucose is converted into pyruvate. In first stage, energy consumption is there, and in second stage energy is released. But in case of gluconeogenesis, pyruvate is converted into the glucose. So in case of glycolysis, if energy is created, so in exactly reversible procedure of gluconeogenesis, where glycolysis release energy, in that stage gluconeogenesis consumption takes place, consumption of energy. So this is exactly reversible procedure of glycolysis. Gluconeogenesis is also takes place into the cytoplasm, and in case of lots of procedures are lots of steps are also included in gluconeogenesis cycle, like your glycolysis cycle. So the first stage of glycolysis cycle is considered as a last stage of gluconeogenesis. In case of gluconeogenesis, pyruvate plus ATP molecule, it forms the oxaloacetate plus ADP plus phosphate molecule, and oxaloacetate plus NADH molecule, it forms malate plus NAD molecule. The molecule can exit mitochondria mainly by conversion to the malate in the cytoplasm. It is changed back to the oxaloacetate. In case of mitochondria, conversion into malate, but in case of cytoplasm, again malate is converted into the oxaloacetate. In case of cytoplasm, malate plus NADH form oxaloacetate. Like this is the exactly reversible step of mitochondria. And in case of endoplasmic reticulum, glucose 6-phosphate is converted into the glucose, catalyze the glucose 6-phosphate reaction, like glycolysis cycle when glucose is converted into glucose 6-phosphate. Importance of gluconeogenesis is to control the blood sugar level, and human system produces glucose. The process of gluconeogenesis in the liver result in the production of glucose, which is released in the circulation for the use of energy throughout the body. Like earlier discussion, energy is released during gluconeogenesis, which is used by the body during the starvation. So that's it. Thank you so much.